Hi friends, today I am going to tell you about a unique and fantastic creatures which are existing on this earth. Those are popularly known as magnetosomes, magnatotactic bacteria, magnetosomes and their applications. And I'm going to cover some of the topics in this presentation where I'm going to cover introduction, important characteristics of magnatotactic bacteria, magnetosomes, mechanism of magnetism formation, physiological conditions that affects the magnetism formation, applications and conclusion. We all know that around 4.6 billion years ago, the life was originated on this earth as a small, tiny unicellular creature. And as the time changes, the life also changed from unicellular to multicellular form. During this, there was a lot of changes occurred in our Earth's environment. In that, one of the most important change that was happened around 1.9 to 2 billion years ago, that is increase in or change in concentration of oxygen. This is because of due to the evolution of photosynthetic microorganisms. And in that, prior to evolution of oxygen and photosynthesis, the predominant form of iron which was existing on this earth was ferrous form, which is reduced form of iron, and it is very easily soluble in waters. So as the concentration of the oxygen increases in our earth atmosphere, this increased oxygen that reacts with reduced form of iron and converts that into a ferric form, that is this is insoluble in water, and that forms a insoluble minerals like hematite, ferrohydrite, magnetite, gregarite and so on. And this is a reaction how the ferrous form of iron reacts with oxygen and leads to form a ferric form, that is insoluble in water. And this change in concentration of available iron may a group of bacteria take up available form of iron and stored in their body as small magnetic iron crystals. And we are in 1963, one of the scientists called by the name Salvatore Bellini, and he observed when he was observing a bag sediment under a microscope, he found that a group of bacteria evidently orient themselves in a unique direction, and later that he realized these organisms moved according to the direction of North Pole and called them as magnetosensitive bacteria. And one more scientist in 1975 and named by Richard Blackmore, and he also independently observed and same thing as the scientists Salvatore Bellini observed, where some of the microorganisms capable of orienting themselves in a certain direction, and these organisms follow the earth magnetic field. And finally he gave the term, and he called them as magnatotactic bacteria, and he also gave the terms like magnetotaxis and magnetosomes. We all know that taxis means phenomenon of momenting with respect to any external forces or any external attractions, wherein magnetotaxis means the phenomenon of movement in response to external magnetic field is called magnetotaxis. And magnetosomes, and whatever the phenomenon means, magnetosomic phenomenon is due to the presence of nano-sized magnetic particles that that act as a small compass needle for orienting these bacteria to the north pole and south pole of the earth magnetic field, call them as magnetosomes. And coming to some introduction about magnatotactic bacteria, these magnatotactic bacteria are ubiquitous. They are existing all over the world, and belongs to gram-negative prokaryotic microorganisms, and they are usually habited by freshwater and marine water, and some of the sediments of both freshwater and marine water, and have ability to passively align themselves along Earth's magnetic field due to the presence of intracellular organelle called magnetosome. Almost all the magnatotactic bacteria are microaerophiles or anaerobes or facultatively anaerobes. Therefore, they are generally found in a region called oxy-anoxy transition zone or oxy-anoxy interface. And we can know what you mean by oxy-anoxy transition zone, where exactly it exists in the water body. Oxy-anoxy transition zone is a water column wherein the concentration of the oxygen is minimum or optimum for growth and development of these magnatotactic bacteria. And you can see here oxy-anoxy transition zone, where the water column which exists above this oxy-anoxy transition zone, which is rich in oxygen, that is anaerobic environment, and where the water column which below the oxy-anoxy transition zone, where it is called as anoxic region and anoxic region. And these indicates the bacterial structures, where if the bacteria is present in oxygen rich environment, they rotate their helical flagella in counterclockwise direction and move towards oxy-anoxy transition zone. And if the magnatotactic bacteria present in anoxic region, anoxic transition zone of our water column, they move their helical flagella in clockwise direction and reach the oxy-anoxy transition zone. And this is how these organisms usually strictly present in or habitat in oxy-anoxy transition zone, where the oxygen concentration is minimum. And coming to one of the important point, that's the most magnatotactic bacterium, that is, or the first magnatotactic bacterium, that is Aquaspirillum magnatotacticum, which was discovered and isolated by our scientist Richard Blackmore, and presently this organism was renamed as Magnatospirillum magnatotacticum. And this is a picture of the Magnatospirillum magnatotacticum. And we all know that all the bacteria which are existing on this earth are having a diverse morphological shapes, like rods, cocci rods means a rod shaped bacteria, cocci means round, and we all know that different shapes which are existing in bacterial group, as like same as that of this, in this group also there are so many different morphological groups which are existing in this group also. In that, important morphological shapes like vibrio, rods, cocci, spirilla, and some of the multicellular or multicelled organisms which are existing in this group. We can see all the type of pictures here. And coming to some physiological tree or different examples of magnatotactic bacteria, and based on 16S rRNA based phylogenetic relationship of magnatotactic bacteria, and the magnatotactic bacteria which are isolated and maintained in pure culture, they are indicated as bold letters and underlined. And the magnatotactic bacteria are classified under six groups, wherein in that alpha proteobacteria, gamma proteobacteria, nitrospira, and magnatotactic many-celled prokaryote. And this is how the phylogenetic tree of magnatotactic bacteria. And these are some of the examples which are isolated and maintaining in pure culture. That's Magnatospirillum magneticum, Magnatospirillum magnatotacticum, and so on and so on. And these are some of the examples as I mentioned in earlier. And coming to one of the most important and core concept of this organism, that is magnatosomes. Magnatosomes are the key component of magnatotactic bacteria, and it is defined as intracellular membrane bounded iron bearing inorganic crystals. Those magnatosomes comprising of iron oxides, iron sulfides, or both. In order to distinguish these particles with artificially synthesized magnatotactic particles, they are usually referred as bacterial magnetic particles. And magnatotactic bacteria are known to produce two types of minerals. One is iron oxides and another is iron sulfides. And the magnatotactic bacteria that produce iron oxides, those only biomineralize magnetite, and the magnatotactic bacteria, those who produce iron sulfides, only biomineralize gregarite. This is one of the most important point about these magnatosomes and biomineralization. And magnatotactic bacteria are commonly analog one, two, or many magnetosome chains, each made up of an average of 20 individual magnetosome crystals. And the Gregarite mineralizing magnatotactic bacteria are usually strictly anaerobic, hence they are found in usually anaerobic environments. And next another is magnetite. The magnetite mineralizing magnatotactic bacteria are usually microaerophiles or aerophiles or facultative aerophiles. And coming to some properties of magnetosomes, the magnetosomes are core. Magnetosome core is usually of high levels of purity and crystallinity, and magnetosomes are usually large single magnetic domain nanoparticles and stable at physiological temperature. The magnetosomes are covered by magnetobacterial or biological membrane made up of majority of lipids and majority of proteins, and magnetosomes can easily functionalize due to presence of various chemical groups at their surface. And these are some of the properties of magnetosomes. And coming to the morphology, size, composition and arrangement of magnetosomes are species specific, and they are chemically, biologically and genetic controlled. And this is how there are so many types of magnetosomes morphology which are existing in different groups of magnatotactic bacteria. In that, usually or commonly existing morphology of magnetosome in different bacteria like cubo-octahedral, bullet-shaped, elongated, spheroidal, then rectangular, and some of the magnatotactic bacteria having either single chain or multi-chain. And coming to the size, a mature magnetosome typically fall within a narrow range of 35 to 125 nanometer size, and the magnetosome in this range are stable single magnetic domains which are permanently magnetic and have maximum magnetic dipole moment. If the size of the magnetosome is less than 35 nanometer, that magnetosome is called as superparamagnetic particles, and I will explain what you mean by superparamagnetic particle in next slide. And if the size of the magnetosome is more than 120 nanometer, are non-uniformly magnetized because of the formation of domain walls, that is multiple domain walls, and hence the net magnetic resonance power or the net magnetic moment of each crystal is neutralized or very less, hence they are not magnetic in nature. Here in this graph you can see that what you mean by superparamagnetism and what you mean by a perfect magnet and what you mean by multi-cell domains or what you mean by non-magnetic crystals. And if I already told, if the size within a range of this 35 to 125 nanometer, they are called as permanently magnetic, because and if the size is less than 135 nanometer, they are called superparamagnetic reasons because they doesn't have any net magnetic dipole moment. And if the size is more than 125 nanometer, the net due to formation of multi-domain walls. Here you can see the multi-domain walls. What you mean by domain means a surface of a magnetic material or magnetic element, and we can see that a cluster of billions of atoms that all have magnetic fields lined up in the same way is known as magnetic domain. And this is, you can see here, all the atoms which are present in this element are magnetic field lined up in a single direction, wherein you can see here there are multiple domain walls. Here in some parts some atoms are existing in one direction and some others existing in or moving in another direction, hence the net magnetic dipole moment is 0. And magnatotactic bacteria produce their magnetic particles in chain usually, and therefore by controlling all particle size and arrangement, magnatotactic bacteria have optimized the magnetic dipole moment per magnetosome. And this is one next concept is how these magnetosomes which are responsible for magnetotaxis are formed in this bacteria, and that's called a mechanism of magnetosome formation. And there are many scientists proposed many pathways or many steps how these magnetosomes formation occurring in this bacteria, but in recent studies one of the scientists studied in 2008. His name is Arkaki and his co-workers. They propose there are four important steps which are existing in formation of magnetosome. The first step is invagination of cytoplasmic membrane leading to vesicle formation. Then the formed vesicles aligned in a straight line. Then transport of iron into cell and then transport that into vesicles. Then finally nucleation and growth of magnetic crystals. Coming to some genetic aspects of magnetosome formation, and we all know that magnetosome biomineralization is generally controlled by two types of genes. One is MAM gene and another is MAMS genes. MAM gene means magnetosome membrane genes and MMS genes. MMS means magnetic particle membrane specific genes. These two genes which controls the magnetosome formation, and there are about 30 proteins which are approximately identified which are involved in magnetosome formation and almost all the genes which are present in four operands: MAM AB, MAM GFDC, MAM XY, MAM and another one operands, and some of the genes which are confined or they show that they are strongly conserved among different species, in that MAM AB, EI, KM, O, P, Q. These are some of the genes which are strongly conserved in different species. And here I am going to, yes friends, what are the genes which are involved in magnetosome formation? There are so many genes which are involved in magnetosome formation, and in that some of the genes which have their specific functions, like yes, the MAM A gene, which is which have a main function. The main function of MAM A gene is activation of magnetosome, activation of vesicle formation is the main function of MAM A gene. And the next gene is MAM B and MAM M. MAM B and MAM M. They are which are exclusively involved in iron transportation. The MAM B which is involved in iron transportation and MAM M which is involved in inorganic ion transportation. And next genes are MAM K and MAM J. The MAM J gene which is involved in magnetosome chain formation. Here MAM J and MAM K is a hair-like or protein-like fiber-like protein which helps in chain formation of these vesicles, and it's also involved in cytoskeleton filament. And next is MAM T. It's a heme binding and reoxidation of iron. Is here MAM T. MMS6 gene which is involved in iron binding, and MMS gene here iron binding and crystal growth, which is responsible for iron binding and crystal growth. And next is MAM N, which exclusively for hydrogen ions, or it excludes the hydrogen ions from vesicles to maintain the pH. And some other genes like MAM C, MAM G, MAM F, MAM M, which are control crystal growth in a cumulative manner. And this is how there are so many genes which are involved in this biomineralization of magnetosome, which is in magnatotactic bacteria. And coming to some physiological conditions that affects the growth or the magnetosome formation. Actually, besides biological and genetic control, it has also been demonstrated that the formation of magnetosome is very sensitive to change in solution chemistry and physical conditions. In that, some of the factors like temperature, pH, external iron concentration, external magnetic fields, nutrient availability or concentration, these are some of the external factors which positively affect the formation of magnetosome in magnatotactic bacteria. And in one of the research paper, the scientists published what are the actual magnetic factors or external factors that affects the growth and development of magnetosome in Acidotheobacillus ferrocsidens. And we all know that this is the introduction about Acidotheobacillus ferrocsidens, and they use some of the materials and methods like Acidotheobacillus ferrocsidens from acid mine drainage, and they use modified 9K medium for growth and development of this Acidotheobacillus ferrocsidens. And this is the composition of modified 9K medium, and they are used different factors at different levels. One is external concentration of iron sulfate, then concentration of ammonium sulfate, temperature and pH. These are some factors at different levels. And in this graph you can see that the effect of ferrous sulfate concentration on growth and formation of magnetosome in acetothiobacillus for oxidants. Here you can see here, as the concentration of the ferrous sulfate increased from 40 to 120 millimolar concentration, the formation of magnetosome is extremely increased. But after that, after more than 160 millimolar concentration, the magnetosome formation is reduced. In this graph, you can see that effect of ammonium sulfate on growth and formation of magnetosome. And next important factor is temperature, effect of magnetosome temperature on growth and formation of magnetosome. That is, as the temperature increases, here you can see that as the temperature increases from 20 to 30, you can see a gradual increase in magnetosome formation. Hence, the optimum temperature for magnetosome formation is, that shows that 20 to 30, and after that it reduces. And this is the effect of pH on growth and magnetosome formation. And coming how we can isolate these unique fastidious growth magnatotactic bacterium from samples. And I already told that these are ubiquitous in nature. They usually habitat in fresh waters and marine water sediments. So we usually use popular method called as racetrack method. Here these are some of the steps which are involved in the racetrack method. First, we have to select the site from which we are going to isolate the magnatotactic bacteria, and usually we have to select a site or suitable places where usually wetlands are more congenial for isolation of these magnatotactic bacteria. And second one is collecting of the sample from a suitable places. After that, this is collected sample. This bottle shows the half. In this bottle, we have to fill half of the sediment and half with the upper layer water, and bring it to the laboratory and keep it for settlement of all the particles. Either it may be a 1 week or 15 days. After complete settlement of all the particles, we have to put two magnets of opposite directions, south and north pole, so which facilitate the movement of magnetosomes towards their respective poles. After that, we have to prepare racetrack. Here this is a method of preparing racetrack. And finally, once we kept a specific pole at a tip of racetrack, all the magnetosomes which are there here, they pass through cotton filter and enter into this small tube. And finally, we use this enriched solution for further isolation and study. And the medium which is used for mass multiplication of these magnatotactic bacteria are usually ATCC medium 1653. That is specifically used for magnetic spirillum growth. This is also called as magnetic spirillum growth medium. And this is the composition. And this is the composition of a Oates mineral solution. And we can how we can make Oates vitamin solution. These are the composition. And coming to last part of this presentation, that is applications, where we can use these magnetosomes or magnatotactic bacteria. So due to their fastidious lifestyle and having a specific structure called as, or specific organ called as magnetosome, we can use in different fields like pharmaceutical industries, biomedical industries, and some industries. Biotechnologically, we can use or exclude these organisms, and we can prepare a microbial magnetometer out of this by using these magnatotactic bacteria, and we can use the magnetosomes as magnetofossils. And we can use these magnatotactic or magnetosomes in drug delivery. Here you can see that this is a blood vessel, and these are the magnetosomes. We can treat or we can bind them whatever the drug which we want to transfer it to a specific tissue. We just bind the drug on this surface of the magnetosome and injected these magnetosomes or treated magnetosomes into the vesicle of the blood vesicles. Then we use external magnetic field to regulate their movement, and finally these magnetosomes reach a specific tissue and cure the disease. And researchers have proposed 4 methods for moving synthetic objects. The first consisting of fitting the particles within a magnatotactic bacteria, and second is loading the magnatotactic bacteria with small synthetic particles, and the third is magnatotactic bacteria pushing a large particles from behind, and the fourth is, which is very useful for large particles, which involves these magnatotactic bacteria while swarming from one side to another side, they move together, and along with that they can carry the large objects. Coming to another application where how we can use magnetosomes in curing the cancer tumors, that's called as magnetic hyperthermia. Here you can see that this is a tumor. They are injecting the magnetosomes to the tumor of the mice, and then they applied the alternate magnetic fields so that these magnetosomes get heated, and that heat leads to destruction of cells in and around that magnetosome. And finally, after 30 days of treatment, we can see that there is a disappearance of tumor completely. This shows that we can use these magnetosomes for treating the tumors, cancerous tumors particularly. These are some applications of these magnatotactic bacteria and magnetosomes. And coming to the last part, that is perspectives and directions for future research. Although the tremendous progress in the study of magnatotactic bacteria and bacterial magnetosomes has been made, there are many fascinating questions that remain to be unanswered. It can be hoped that there will be a breakthrough in the following aspects, like the first one is challenges associated with the mass production of magnetosomes need to be addressed, because we don't have that much scientific technology for mass production of magnetosomes till now. Second one is the synthetic and concerned effect to study the functions of various magnetosome genes and proteins which are involved in magnetosome formation in vitro and in vivo conditions. And it's very urgently required, because we can transfer the specific gene to another bacteria and other organisms where we can produce magnetosomes there also. So for that we need a specific function of a specific gene. So this is a second important constituent. And the third is magnetism of magnetosome will hopefully provide useful information about the rock mineralization theory, the acquisition mechanism of the depositional remnant, magnetization and environmental mechanism, because we can use these as magnetofossils. And these are future perspectives and direction for future research. And so I conclude that new technologies are evolving rapidly, constant search of development of new therapies, processes and surgical to overcome all the challenges, difficulties facing in many fields of science. These fascinating little bacteria, teeny bacteria, have opened up new dimensions in many fields of science, and in years to come will be a great importance in future. So we have to start thinking about this fascinating magnetotactic bacteria in your dreams. And bye, and thank you for watching this video. And don't forget to subscribe. Thank you one and all who are watching this video.